This Week in Virology, the podcast about viruses, the kind that make you sick. From Microbe TV, this is TWiV, This Week in Virology, episode 792, recorded on August 10th, 2021. I'm Vincent Racaniello, and you're listening to the podcast all about viruses. Joining me today from Ann Arbor, Michigan, Kathy Spindler.
Hi, everybody. Here it's 86 Fahrenheit, which is 31 Celsius, and it's a pretty hot day. No clouds in the sky.
Here it's 28°C, completely clouded over, but really humid and warm outside. Also joining us from Madison, New Jersey, Brienne Barker.
Hi, great to be here. Same weather here in New Jersey. 82 Fahrenheit, 28 Celsius, very cloudy.
Last week, your weather was like Michigan's weather, right?
It was. I think actually last week I might have had the highest temperature beating Rich, and that was just very shocking.
That's shocking. Austin, Texas. We have a guest today who is returning for a second time. He was here just over a year ago. He's at the Mailman School of Public Health here at Columbia University. Jeff Shaman, welcome back.
Thanks for having me, Vincent. Pleasure to be here again.
We have had a demand from our listeners to have a real epidemiologist back to explain transmission, spread of viruses in populations, R-naught, blah, blah, all that stuff that we don't know because we're virologists. So here we are. And we did talk about some of this last time, but over a year ago, things have changed. So before we start, please tell us, what is an epidemiologist? What do they do and how do they do it?
Well, honestly, epidemiologists are a broad range of people who study health and disease at population scales. Mm-hmm. They use a whole variety of techniques. Some of them are survey related. Many of them are statistical. It's a whole branch of statistics that's really epidemiology, or a branch of biostatistics, if you will. And there are also people more like me who do mathematical modeling of systems. In my case, I do a lot of modeling of infectious disease systems, mathematically and in combination with statistical systems. The idea of epidemiology is to understand the characteristics of diseases at population scales. And it can be everything from something descriptive to like what are the rates of heart disease as you go through different comorbidities or age classes or populations geographically, to, what is the manifestation of how a disease spreads? Why is it able to critically get around? Why are some diseases successful, infectious diseases, and others not? And those are the things that we like to characterize and try to understand and use them to do all sorts of things, of course. Think about interventions, model them out explicitly, make projections and forecasts, design interventions in ways that actually will benefit the population at a population scale.
And would you say that like when I say do an experiment, I'm thinking of I go to the bench, I have an ice bucket, I get tubes out of the freezer, etc. When you do an experiment, what does that entail?
Most what I would think you would call epidemiological experiments are not going to be that kind of core controlled laboratory environment experiment where you really can dive down and control one thing or another. Very often, it's analyses of large data sets that are gleaned from public records. You could take a Medicaid data set and look for rates of heart disease in that. And what are the factors that are associated with it? Be it comorbidities, as we talked about, be it environmental drivers, being life history. You're going to explore this, but you're going to do it with a big data dive. And so instead of being something where you have an experiment that you can control, you really are looking for data sets that you can interrogate in a systematic way to allow you to elucidate associations and hopefully some causal associations.
So in the current pandemic, an epidemiologist could be called on to say, figure out what interventions might do, what numbers might be like at different times of the year, those sort. And so governments could have an epidemiologist or multiple study that, right? And
they do. When you go into, New York City Department of Health and Mental Hygiene or state public health agencies, other municipal public health agencies, they have epidemiologists as some of the people who are employed there, who are doing the analysis to try to understand what's going on. Some of it can be very descriptive. How many people were infected last week? That's a core of epidemiological data. In the case of COVID-19, we want to know how many new cases are there this week? How many hospitalizations? How many people in the ICU? How many people died? What were their age ranges? What were their comorbidities? What were their life history? Had they been vaccinated? Had they previous exposure history? That's basic information that you can then use to understand other things as well, which is, how many people actually are infected out there? We're only seeing the confirmed cases. How many people are truly infected? How rapidly is this virus moving through the population here? Is it growing? Is it abating? Is it got the potential to grow to a certain degree? What kind of intervention might we employ, and what's the likelihood that it's going to be successful? Some of them we can pin down better than the others. That last example I said there, that's a hard question. How good is an intervention going to be? How much reduction of cases will we see in the coming week if 50% of the people will wear masks? Very hard question. We don't have answers for something like that. But there are other things that we can actually really pull apart and tease apart. And that's the type of stuff that in a pandemic, obviously, you really want to know in real time as you're trying to make decisions about how to control it, particularly through non-pharmaceutical interventions, which obviously is all we had really in 2020.
Would you say that running a clinical trial for a drug or a vaccine is an experiment in the epidemiologist toolkit, or is that?
Yeah.
Okay.
I would. So it's not like they're not involved in experimental design and they're not thinking about those things at all. It's not going to be in a laboratory setting, but that what you're getting at there is exactly the type of experiment an epidemiologist might do. Now, they also might go out and do surveys and collect data that way. And there might be ways to control the experiment and the results they're trying to find in a way that secures power and does it in a statistically rigorous fashion as well.
So you mentioned that part of epidemiology is looking at the relationships between different types of data. Mm-hmm. And you said that sometimes you hope to find something that's a causal relationship. Mm-hmm. How do you sort of just say that a relationship is causal? What makes
that's a slippery slope. And I actually have had problems with that also very much. There's a whole branch of statistics and biostatistics and epidemiology dedicated to causal inference. And I am not going to defend it or denigrate it right now on this podcast. I'm going to let somebody who's a proponent of it come on another podcast and talk to you about it, because I have my issues with it as well in terms of what it can and can't do, and whether it's really being able to establish causality. And I think actually somebody who's more informed of those methods would be better. You would be better served listening to that person rather than me.
Okay. Yeah. Causality is always difficult, even when you have all the players lined up, right?
Yes.
But for example, in a vaccine trial, you always have an epidemiologist in any kind of clinical trial, right?
Yeah. Yeah.
And then if there's some rare cases of myocarditis, then they can start to say whether that might be causal or associated, right?
Well, they would say whether or not it's, let's say, statistically aberrant, whether the evidence supports that it was highly unlikely that that level of problem arose strictly by chance.
Okay.
And so when you reach that level of statistical significance, and that threshold is somewhat arbitrary, but when you get to that point, then you really have to take pause and say, okay, what is going on here? We have something that's anomalous. It doesn't look like it arose strictly by chance. What do we need to find out about this?
And if some side effects, say, happened always 14 days after the immunization, that would give you some clue, right?
Yeah, because if it happened over and over again, it would probably very quickly rise to the level of statistical significance. And you'd say to yourself, there's something happening with great regularity here, which is not what we would like to see happening. And we're going to have to figure out what it is. And we've seen that already, with some of with the J&J vaccine, and I think AstraZeneca, they had pauses on it. And it's certainly possible to argue with, well, was that really justified, or was that just intended to mollify any sort of public hysteria that might have come about? And in the case of the J&J one, it was very interesting because, you look at it and first blush, it was some kind of form of stroke, if I remember correctly. And first blush, you say, well, look, birth control pills induce a higher rate of stroke than this vaccine is by quite a lot in women. And it was mostly in women that we were seeing this. But then when you bored down, you saw it was a very specific, unusual type of stroke. I don't remember quite what it was. And that actually, to my mind, made me say, all right, let's give this pause and figure out what's going on here. Even though this is rare, they're all aligning with something that is incredibly rare, much rarer than this. So there may be something there that's going on that requires an adjustment of who you're willing to give that vaccine to.
So I wanted to talk about the movement of viruses through a population, which has been called transmission, contagion, I don't know what else. And since late last year, actually since February 2020, a series of variants have arisen, starting with D614G, and then at the end of the year, alpha, beta, gamma, delta have arisen, each being called more transmissible than the last. And now we're at delta here, which appears to be the uber variant of all. So I wanted to talk about how we study this and what are the metrics that we use. So I as a virologist, and perhaps Kathy and Brianna are familiar with reproductive index, secondary infection rates. Are those the ways that we measure a virus's ability to spread in a population?
It is. But again, we would be looking at it at population scale. And, there's a blurring. There are people who will do household infection studies of influenza, and they'll look and they'll see what the secondary attack rate is within the household. And they'll use that to try to understand influenza and its transmission within the community. It's a very specific instance. So it's important when you're looking at any of these studies that you look at what's the context in which they did this. What were they looking at to actually come up with that number, to diagnose it? And of course, what are the methods that they use? Now, for this disease, when it came out, one of the first things people wanted to know was, what is the reproduction number? Sometimes they would call it the basic reproduction number. I think it's best if I give you like 101, my take on all this stuff. All right. So the basic reproduction number is called R-naught, and it is strictly defined as the number of secondary cases an infectious person would cause in a fully susceptible population. So you can imagine at the very beginning of this outbreak, it's emanating from the epicenter in Wuhan, and people are getting on airplanes and they're going to Seattle and Bangkok and Tokyo and Seoul, right? And they're seeding infections with this novel agent. The average number of infections they cause would be called R-naught. Now, people have traditionally looked at this, maybe dogmatically, as an immutable number, but it's not, never has been. And that's one of the really critical things to understand. So if you think about it, if you were to plunk a person down in Manhattan, New York, on an average pre-pandemic day, when they're getting on the subway and public transit and they're going to restaurants and they're in this densely packed island that swells to almost 4 million people during a typical workday. That is very different if you compared with plunking them down in eastern Montana, in a highly rural ranch space community where people only interact with relatively few people compared to Manhattan, and it's a car culture. These are structural differences that have to do with population density, how much people interact, how much communal transportation work there is. All those factors are going to affect the likelihood that somebody has the opportunity to transmit to someone else. The more people you encounter if you're infectious, the more opportunities for transmission there very likely are. Now, because of that, we would expect that there are differences in that basic reproduction number as you move from location to location, based on those structural differences that exist. All right. Now, we could still call it R-naught if you want, but it's getting slippery already, because what that means is that the reproduction number in Manhattan is not the same as rural eastern Montana. And it's likely not even the same as Seoul, South Korea, even though they're big cities that have the same kind of, public transportation systems and they're dense. There probably are differences there. So when this disease first emerged, you heard estimates of it, and I can get into how we do those estimates in a moment. and you get different numbers. And those numbers, in part, reflected the different locations and populations people looked at. They also reflected the different methods and the time points and the quality of the data that were being used to try to make that estimation. Now, R-naught can differ structurally from space, but it also can differ in time. And at that point, I would stop calling it R-naught and I would call it RT, the time varying reproduction number. And it varies in time for a number of reasons. One, some viruses have a sensitivity for reasons that are understood a little bit or not to environmental conditions. So for instance, with influenza, we have a sense that it's sensitive to both UV radiation and humidity conditions. And we have less of those in the wintertime. And that is favorable to the survival of the virus once it's expelled from the human host. And that seems to allow the virus to transmit better during the wintertime and not as well during the summertime. So on top of that structural difference from place to place, you're also now getting a temporal envelope, a movement where it's less transmissible in the summer and it's more transmissible in the winter. So that's one thing that can affect it through time. Another thing are what people do. Typically we haven't had to think about this when we're thinking about diseases like flu outside of a pandemic context. But with COVID-19, we disrupted our lives. We had lockdowns. We wore face masks, more or less, depending on the location and with different efficacies and rates and cloths and, putting it on your chin, putting it over your face correctly, et cetera. We socially distanced. We shut down schools. We shut down a building, office buildings. All those things are limiting opportunities for transmission to better or worse degree. Some of them are effective, some of them are not very effective. But all of them are changing that reproduction number. Because if you go in the same population and everybody's masked, the rate at which the virus moves through that population is going to be slower, right? Which means that the average person is going to infect fewer people than if everyone were unmasked. All right, so we have R-naught, which is this ideal of how transmissible the virus is if you plunk somebody into a fully susceptible population. Then we have this recognition that it changes from place to place because population density, use of public transportation, et cetera, those features change. So it's not the same from place to place. In addition, it changes through time, some of it because of regular environmental variability that affects transmissibility of the virus, and other things that are much less regular that have to do with human behavior. And very often that's a very minor effect, but in this pandemic, it has not been. Those lockdowns, the masking, they are actually having a substantive impact. Now, on top of that, there is a third reproduction number we have to talk about, and that is called RE, or the effective reproduction number. And that takes into account that, in truth, in most populations, not everybody is susceptible to infection. Some people have been vaccinated and it's conferred sterilizing immunity. Some people have been previously infected, and that too is precluding transmission, at least temporarily, right? And so if you really want to know the thing that most people are estimating, is they're getting an estimation of the effective reproduction number, which is that RT multiplied really by the fraction of the population that is susceptible to infection at that time. If that number is greater than 1, it means in that population at that time, the average person is transmitting the virus on to 1 or more people. 1, if it's equal to 1, greater than 1, it's more. If it is less than 1, the average infected person over the course of their infection is transmitting to fewer than 1 person. And as a result of that, if it's less than 1, the virus will locally die out. The number of cases will decline if that were to stay in place. If it's larger than 1, it will continue to grow as long as it is larger than 1. But for the effective reproductive number, it's also important to remember that as time goes on, you are burning through and using up your susceptibles. So any outbreak that occurs is self-limiting. And it's self-limiting because you start to run out of as many susceptible people in the population. Now, you could imagine that you have a reproduction number that's greater than 1, and it allows the average person to infect 2 people. And those 2 people could then infect 4 people and so on. But the next week, because susceptibility is a little bit lower, those 2 people are actually not going to infect on average 4 people. They're going to infect a little bit less because we already used up 2 of the people. And as that goes on, that slows the growth of the outbreak down until that reproduction number, that effective reproduction number, hits 1, at which point that's the peak and it starts to come back down.
So in effect, the reproduction numbers that you're telling us are not completely reflections of the intrinsic transmissibility of the virus, right? Whatever viral properties are needed to get it from one host to the other. It's many other factors all lumped in together, right?
Exactly. It is a combination of all those effects. Think about it. The effective reproduction number, which in the here and now is the most important thing. It tells you whether or not you're going to see more cases tomorrow and the next day and the next week. Okay. That is a function of the susceptibility of the population, of the NPIs that are in place, of the seasonality and its impact on the virus, of structural differences in population density and whatnot that affect it, and those intrinsic characteristics of the virus.
Now, originally you said early on in the outbreak, the R-naught was calculated, which, as you said, is not really a constant. But we see these tables in textbooks. My textbook is guilty of it. The virus R-naught. I mean, what are we to make of that? I mean, they could obviously they can.
Yeah. I mean, what you would hope is that a couple things. Firstly, you have to know that there's uncertainty around it, right? Like anything that we deal with, there's uncertainty around it. If they tell you this has an R-naught, well, somebody tells you it's got an R-naught of 2.46385. All right, that's way too many significant digits, okay? That's a little nuts. If they tell you it's got an R-naught of 2.4, really you should be saying to yourself, well, that's maybe if they did this well, it's somewhere around 2.4 for what they said. Maybe it's between 1.8 and 3.2 or something like that, okay? You would love to see the uncertainty of that estimate as well, not just the number. And what you often saw, and we saw this at the beginning of the COVID pandemic, is you'd see a range of estimates for R-naught. And they'd be from different locations and different groups using different methods. And you'd want to see where they were. And you'd see that most of them lay between 2 and 3. There were some outliers that were higher up, but you were sort of getting a consensus from people doing their analyses on different populations with different quality data and different approaches that they were making estimates. And they were sort of aligning in that area. But as you say, does that mean that that's how it's going to behave if I take it? If I go in the middle of the desert, nowhere, I can't infect anyone, right?
So to paraphrase the pirates, it's more of a guideline.
It is absolutely more of a guideline. Now, the other thing is in the table, what's actually really important is if all those numbers for those different viruses were attained using the same methods and roughly the same general populations, then you have a basis for comparison. And you can look at measles and go, well, this is much more contagious than influenza, right? And you can get a sense that, what it would really take to control a measles outbreak versus controlling a flu outbreak.
Go ahead. Sorry.
So I was also thinking about those tables as you were talking about this. And I have another question about them. And this may be a question that's not really answerable. But are they really what you would consider an R-naught in that table? Because how have we measured some of those things in a completely susceptible population?
Well, that's a really good question. But the reality is actually there are a couple ways of disentangling the susceptibility of the population. And that's actually not that bad. I think the better question is it really an RT or is it an R-naught?
Sure.
How much have they controlled for seasonal differences, the populations they looked at? Was this all based on urban data? What does it mean for a setting that's not urban? That's really important to think about and know, and that's what you'd like to distill. But there are a couple ways of estimating R-naught. or RT, let's say. One is to look early in an outbreak, like right at the beginning of the pandemic, and you can expect a rate of exponential growth based off of simple forms of compartmental models, the SIR type model that you've heard so much about. And you can actually linearize that and make it an estimation problem using just a regression framework and a certain assumption that the population is wholly susceptible, which is justified if you're very early in a pandemic. And then when you do that, you can make an estimation of RT. In my group, we use mathematical models coupled with inference. And in that, we are able to disentangle the susceptible portion and that RT from each other. So on a daily basis, we can make an estimation of how transmissible the virus is in a locality, putting aside our estimation that maybe only 40% of the population is susceptible right now, or whatever it is. So we can actually disentangle it. There are a number of quantitative methodological approaches that allow you to separate that. The harder part is, well, RT is now whatever it is here. What would R-naught be? In the case of this pandemic, I don't know how many people are really wearing masks. I don't know if they're really getting together with their normal proclivities in the normal ways they do. And what does it mean? I mean, if this is all declared over, is it going to be the roaring 20s and everybody's going to have these enormous parties maskless? Well, then the R-naught that we might have estimated wrong is going to be too low because people are just allowing it to spread wild.
So last week or the week before, the CDC released this chart, which was originally in the Times, of R-naught, and they superimposed Delta. They said R-naught is now like chickenpox. So from what you said, that's not correct, right?
Well, it's higher. It's higher. And, that box is pretty big. Yes. Chickenpox was a point, and the box was pretty wide, you'll notice there. And so what I think, firstly, I don't think the CDC wanted those slides leaked initially the way they were. Secondly, it was internal document. But that box, the width of it there on that x-axis is really, I think it goes from, what did it go from 3 to 6 or 4 to 8 or something like that? That's a big uncertainty range right there. And I think what you're getting there is that the real thing to look at is that it's shifted to the right of where the ancestral lineage is. And, look, in our own research, we have done estimations of how each of the different major variants that we've seen, alpha, beta, gamma, and delta, have resulted in changes in transmissibility upon their emergence in the countries of origin that they're associated with. We don't know where they emerged, obviously. So for alpha, it's the UK. For beta, it's South Africa. For gamma, it's Brazil. For delta, it was India. And this is work that was led by Dr. Wan Yang, who's a professor in epidemiology here at Columbia. And she built a system that allows the disentanglement of, and it's an attempt at it. It has uncertainty, so don't think that this is precise, but how much more transmissible did the virus become at that time point when it emerged and the sequence data shows that it's there? And in other words, how much did it ramp up? And is there actually evidence that people were being reinfected? And actually what she was able to find is very consistent with data that was found using other methods, that the alpha variant is about 50% more transmissible, but was not really causing repeat infections. That the beta variant was only marginally more transmissible than the ancestral line, but appeared to be causing repeat infections. And this is evidence we also see from its ability to evade some of the vaccines that have been developed. And for Delta, unfortunately, it's a combination of the two. And that she showed that it's even more transmissible than Alpha, but it's like Beta, it's also able to evade immunity. Might not cause severe, critical, and fatal disease for people who are having these breakthrough infections, but they're getting it at a higher rate than they were with the ancestral lineage.
So these are attempts to dissociate all the environmental population factors that we've discussed from the virus itself, because as we know, these variants have different genome mutations that are thought to be associated with some increased ability to spread. So you're trying to separate that, and you think these percent more transmissible, I think you put it, reflects just the virus part. Is that correct?
We think your question is right on point, and it's totally from your perspective, Vincent, which I love actually, which is that, when you're thinking about it, what is the mechanism? What's actually going on here? And what we're doing is we're working for the other end. We're saying, all right, we have this information about cases. We see the time series as it moves along. We know what the weather conditions were a bit, and we're going to attempt to control for seasonality. We know that there wasn't any uptake of vaccine yet in those populations for the time periods we were examining. What can we infer about the changes that took place in the virus right at the time that this thing emerged and overtook the ancestral lineage and became the dominant strain? In those 2 months periods, how do its epidemiological properties change? How did RT change? And you're right. Is there an assumption there that, well, suppose they had a lockdown in the middle of it? Well, we try to control for that. And that is a problem for the analysis that we had for the UK with the emergence of alpha. They were doing that at the time. India, for instance, didn't have that at the time. But what we're estimating there is some combination of change in the virus's properties and maybe changes that are due to seasonality. We try to control for that. Maybe changes due to broad differences in mask usage or lockdown policies. We try to control for that if we have information. Some of it we do, some of it we don't. So what we're trying to get at when we do that is this virus moving through the population faster?
Now, Ron Fouché, who was on a couple of weeks ago, says that this is the moving through the population is actually a reflection of the fitness of the variant, which is, how well it moves, how well it reproduces in its host, right? And he says influenza viruses, variants arise every year that are more fit. They displace previous viruses, but they're not more transmissible, and no one ever calls them more transmissible. So he questions why we are doing that for these variants, right?
I think that's an interesting point, but I think maybe the distinction there is this is a novel emergent virus that has not had the time to evolve to its capacity to be transmissible. Evolutionary biologists will argue that with serial passage, you're going to select for something that is more fit and more capable of being transmissible. What am I trying to say? Transmitted. Thank you. Transmitted. And I think, the question is, has this virus reached its better potential for moving from host to host, its best potential for moving from host to host within humans? And I think that's what we're seeing the difference of. And his point about flu, I think, is spot on, which is, well, flu has a really pretty low reproduction number. It's generally estimated between 1 and 2, It's not that great. Why hasn't it taken the time to make itself better? Why hasn't it made itself more like measles? Well, of course, I'm anthropomorphizing it. The virus doesn't have an agenda. And maybe that's the limits of what that virus is capable of, given its receptor binding, given its ability to withstand salts and pH changes and droplets it's expelled, all the molecular processes that are at play. But maybe for this virus, it hasn't gotten there yet.
Yeah, I think that makes sense. I always looked at it, this thing ran out of China in the beginning, and it was pretty damn good at traversing the world. But that's from our perspective. Maybe it could get better, or maybe it could very well be that the drive through the population is a consequence of other changes that are selected by other forces, not actually moving from one person to another, and they're incidental changes. That, I think, virulence is often an incidental change, a consequence of something else, because to me at least, it doesn't make sense to increase virulence for a virus. But
I'm with you there, Vincent. I don't, there's some arguments that actually evolution will select for less virulence. I don't buy those either. I just think it's kind of floating around and sort of piggybacking on other things. But, one of the, I think, outstanding questions that we're really going to have to understand is, what are the selective processes going on both within hosts and between hosts? What are the bottlenecks that are allowing a variant which emerges? And we've seen this in people who are immunocompromised. People have HIV, have lupus, cancer therapy, blood cancers. individuals who are not able to mount a response, they have these very protracted infections with COVID-19. And they've sequenced them, some people 20 times over the course of an infection over 100 days. And you can see the diversification that's going on, and then the crashing of it. And how, they'll put them, they'll give it one of the monoclonal antibody therapies, and it's this enormous selective pressure for new variants that arise. And then when it's taken away, it diversifies again. And sometimes it doesn't what's called fix, and it goes back to the old assemblage that was there. That's just better at dealing with the host's body in the absence of that monoclonal antibody therapy. So, teasing all this out and understanding where the selection is taking place and what the potential this virus has in terms of, I'm with you. I don't think as much in terms of virulence, and I hope we don't see a more pathogenic strain by chance or because of some selective force in the future. But what is its capability in terms of how transmissible it could be? That's something we need to figure out.
So I think a lot of the selection is immunological selection. We see all these spike changes, but what's been ignored are the changes in RNA polymerase, the non-structural proteins, other genes could be doing all sorts of things, and that needs to be looked at in the lab. It's the only way we can do that. And of course, it's too soon to be able to do that. But as you said, epidemiologists have to work in real time and get the data out there. I guess what I object to is the news say this virus is 90% or 60% more transmissible when there are clearly other factors at play, and they have not done the work that you have done to dissociate them, right?
Yeah. But I think what you're dealing with there is, something that's difficult. It's been very, very challenging, the communication regarding this. Look, let's look at some of the positives. The positives is people know what a virologist and epidemiologist are now better than they ever did, right? There's actually some level of science literacy in these areas has really been elevated, which is really nice to see, even though it's a really unfortunate circumstance to bring it about. But there are clearly been issues within science that I still think are really, really troubling. And they have to do with the inability to appropriately recognize when you're not nuancing something sufficiently and overly nuancing other things, maybe. And having the right people there to steer that conversation and can sensibly message what we need to message. I'll give you a couple examples. One example was very early on, I heard the WHO in early 2020 saying that we don't want to recommend face masks. We don't think the evidence is good. We think people are going to play with their face masks. They'll touch them all the time. And by touching the masks, then they're going to touch their face, and it's going to lead to more transmission. It's a nice narrative, but where's the evidence, right? I mean, that's all I could say is there is some evidence that masks work, okay? Even then there was, and there's some more evidence that they do, but it was just sort of a narrative that clearly people had repeated to one another that made sense on some level. Another was droplets. Very early on in January, February, I heard people proclaiming it's transmitted via droplet. It's not transmitted by aerosol. And some people would point to the reproduction number and say, well, if it's aerosol, it's going to be like measles. And I would just say to myself, who said anything about a correspondence between R-naught and the mode of transmission? We don't know how flu is transmitted, which is the most studied respiratory virus there is. How could we possibly know how this novel entity is transmitted? Why are we telling people one thing so emphatically in the absence of any information? And it was very, very strange to see that dogma and the amount of discourse that had to go on, a lot of it in public, before people would just say, okay, let's be a little more agnostic here, and let's try to build an evidence base and accept that there are things that we don't know. I think this is part of the whole thing, where there are aspects of what we need to do that require getting beyond dogma. And sometimes it has to do with the fact that people with prominent voices, because of their position in public health agencies or on media, and I'm not pointing my finger at you, Vincent, or at the WHO, and I'm not pointing my finger at anybody specifically, they're the voice of reason. but are they necessarily the person who's the expert in everything that we need to really understand about this and have really spent their time devoted to looking at it? And the answer usually is no. And we need to unearth those people and bring them out and shake the information out from them and start doing the research through them to get that going so that we can actually put out sensible information that's appropriately qualified.
So what's the transmission? Can an epidemiologist tell if it's droplet or aerosol or both?
No. You'd have to look at it. I don't think there's any way to do that. I think that to me, the evidence that I've seen right now is they've done some survival studies. They've seen how it survives in droplets. They've seen how it survives in aerosol. Does that tell you anything about transmission? No, we still don't know it. The only thing that we could look at, maybe from epidemiological evidence, is when you have certain super spreader events. When you see that people sitting in a restaurant at all these tables seem to have picked it all up from somebody who was an index case there, and that they didn't have interaction and that they were outside the range of where we would normally consider droplets. Then you have to say, well, is it a conduit that's a waiter, or is it aerosol? Or the church choir practice in Washington State, where 60 people were infected over a 2-3 hour practice rehearsal, and they were spaced out. To me, I look at that, and that really has me thinking, well, maybe there is a role for aerosols here, And so I would again just stress, there's no correspondence between R-naught and whether it's aerosol or not. I mean, if you ask me, I think flu is probably mostly transmitted by aerosol. I don't know this, and all I can say is it's my own hunch, and from having read things about it and been in workshops where people were yelling at each other about it. But it's got a low reproduction number because there are other intrinsic properties of the virus that may make it less transmissible than measles is, even if it uses aerosols.
So when you were on TWiV 607, you said, at the time, because we didn't know the mode of transmission, it was hard to speculate on which mitigations were actually the key ones. And you drew parallels several times to Korea and the various things they were doing, but we didn't know. And so it sounds like you would say that we still don't even know that for influenza. But I'm just thinking back to this past year when the ILI cases were so low, and what was the difference between that and other years? But then I guess we still don't know. Was it the distancing, the lockdowns, the masking, or all of them?
I think that's a great question, Kathy. I think the issue is when you're looking at that, and if back in April when I was last on, if people are talking about droplets are things that fall out. They have some distance that they go. And people have started to debate the distance. And, initially you heard 2 meters, 6 feet roughly. And then people say, no, it could be up to 20 feet. And, how much are we going to space and all that stuff. But if you're doing that, it's also something that you could argue not to mask up about. Okay. Because it says that, well, if you just keep apart, far enough apart, you'll be all right. These larger droplets are the things that have the virus. They're the only ones that have the infectious dose that's going to be adequate to actually set up an infection in a new host. And if you're far enough away from other people, you'll be great. And the reality is, facetiously almost, you don't want to get COVID, stay away from people. If you can perfectly isolate yourself, that was the solution. But we can't do that. If aerosol is there, that's not an option, right? Because suddenly you can go into a grocery store and somebody was there 10 minutes ago filling the air with virions, and they're floating around, and you come in and you inhale them, right? And that can set up an infection potentially, right? So maybe at that point, if we don't know, masking is an appropriate thing, because that idea that you play with it, I got to tell you, my experience in looking at the world and my family and everyone around me is you forget it's there after a time. Usually you're not too aware of it, and people don't play with it so well. And I think it is an effective strategy. So, do I have any ironclad evidence for this? No. Can I tell you the mode of transmission still about this? No. We have not gotten closer to those things. Are these important issues? Yeah. Are they somewhat pedestrian? Some people think so. Yeah. But I actually think it's quite important. We got to figure that out. not just for this virus, but for respiratory viruses in general. Now, the amazing thing, of course, has been the lack of a flu season, which tells us, all right, flu, we do estimate, has a lower reproduction number and is less transmissible than COVID-19. And the measures that we put in place to control COVID-19, though inadequate to stop that massive surge we had in the US in the wintertime, and many other countries experienced it as well, were sufficient to stop flu. Now, is that everything? No, because there might be some competition between viruses, lots of other issues, of course. But it's probably a portion of it, yeah, and probably a large portion, I would bet.
Before we let you go, Jeff, can you tell us secondary infection rate? How's that differ from R-naught or RT or RE, and how do you figure it out?
I think you've got to look at how somebody is actually specifically defining that. So if it's defined, again, in like a household study, then what they're looking at is, all right, we grabbed somebody, we found them while they were infectious. We know they're an index case. Now we're going to find out who else in the family or the workplace gets infected. That's the secondary infection rate. It's actually related to the reproduction number. It's just a slightly different measure. And I think the critical thing for that and for any of these things, R-naught, RT, RE, I hear that I'm sure it's dizzying. And me sitting here waving my hands and talking about it without writing just down those terms makes it more challenging to even follow. But it's really important that you define what you're talking about. A great example will be, we're hearing all this stuff about breakthrough infections by Delta against different vaccines. Let's pick on Pfizer. There's an article in New England Journal of Medicine that said, from out of the UK, that it's still 88% effective. And you got to look carefully and see that it's 88% effective against symptomatic infection, right? And you got to look at those differences. There's another one out of Israel that says 64%, and then it said 39%. But that one says infection, I believe, if I've got this right. All right. So you got to look at that. You got to look at, and then it'll say, but for severe, critical, or fatal disease, which is a completely different category now, you're talking about hospitalization, ICU, or death, it's very effective. All those things, if you don't look at them, it becomes a real mess. And it's already confusing enough, and there's already a lot of disagreement in terms of what the results are that we have to reconcile. How much breakthrough there is the same level thing with these uncertainties as there is with the R-naught. So it's really important to know what the terms are that you're trying to define in that context, because I got to tell you, different people will do this differently. I may call it the time varying reproduction number. Somebody else may not like it. I may call it the basic reproduction number, and somebody else will go, no. It's the basic reproductive number. I actually don't care what you say, because I'm going to get it either way. But I have been corrected on that in both directions, by the way. Somebody tell me it's not Shun, it's TIV, and somebody tell me it's not TIV, it's Shun. I really don't care. As long as I'm defining the term and we're all on the same page, we can do okay. But it's important that we do that stuff.
Well, we could talk another hour, but I know you have a heart out, so maybe we'll get you back in a couple of months, okay? Because I have many more questions, and you're the one to answer them. But anyway, Jeff Shaman, Columbia University, Mailman School of Public Health. Thank you for your time, Jeff. Appreciate it.
Absolutely. Pleasure being here. Thanks for having me, Vincent.
Thank you so much. I learned a lot.
Yes.
Thanks, Brianna. Thanks, Kathy. Take care.
Bye-bye.
Bye-bye.
Bye.
Okay, debrief. Thoughts? Kathy, you have any thoughts?
Yeah, we didn't really get to talk about his preprints, but he did talk about the data, basically, that by doing this, I already forgot the word, disconnection of some of these aspects of the data, they can pull out and say that some of the variants seem to be more transmissible.
Yeah, it's the intrinsic as opposed to all the other things that are. He thinks he can dissociate them, right?
Dissociate, that's the word. Yeah.
But of course, it's a statistical model, right? And
well, I think
not be right.
The word to him transmission means something different than the word transmission means to a virologist.
Sure. But he is trying to dissociate the population effects, the environmental effects from the virus itself. And in the end, he thinks his models tell us that the virus itself is actually more transmissible. And aside from all the population effects, that's what I got out of that, right?
Yeah, absolutely. But again, even with that, there are still a lot of things that he would be lumping under the virus effect. It's sort of how the virus interacts with one individual host is to him the virus effect, and a virologist is going to have more distinctions than that.
I mean, my question is, do the 18 genome changes, let's say 4 of them, just to simplify it, do 4 of them increase the transmission? Or is it entirely other things? I mean, from what he said, it's a model to dissociate the virus from the rest, but I don't think it proves it. And I think you need experiment. Fouché would say you need lab data. And let's say you take Delta into the lab, you do respiratory epithelial cell reproduction, and you find a more infectious virus shed, faster kinetics, whatever, that would be consistent with it. And I would say, okay, it's consistent with his findings. But what if you didn't find any difference? No yield difference, no kinetic differences, then that would say maybe the models are not right. So that's what I'm waiting for. That's why I think to say this is more transmissible is not conclusionable at this point. Is that a word? Conclusionable?
Yeah. So I guess where I would draw a distinction is that, to me, spread and transmissibility are 2 different sides of the same coin, and talking about fitness is just another way. So to me, I don't make a distinction among those, and I see where he's saying we take the data and see what the virus is doing in the population, and we dissociate out these various things. And when we've done those various dissociations, we get to a point where it looks like this variant is more transmissible by this factor. This other variant is more transmissible by a lower factor. I mean, they're not finding the same thing for every variant. And they're also not finding the same thing with respect to reaction with the vaccine or natural immunity. And so I think that lends credence to the fact that there may be something different about each of those viruses that are leading to that. And it could be the constellation of mutations.
Could be, but I'd like to see it shown experimentally. That's all.
Well, and I think this goes into what he was saying about definition of terms. He has disentangled the sort of what he thinks of as what the virus is doing, the intrinsic parts of the virus, from what's going on with the people.
Yeah.
We could do the exact experiment that you propose and look at how the virus reproduces in culture. And you might say, oh my gosh, there's no difference in this particular model. And you would say, okay, the model and the data, the experiment don't line up. But maybe the difference is actually in evading innate immune responses in the nose. And that wasn't taken into account in the experiment you did. And so in both cases, it's sort of a question of how do you define this difference in transmission?
Right. And you could also say, you did this experiment in respiratory epithelia and you didn't see the difference. Well, how do you know you had the right respiratory epithelia? How did they have all the goodies that they needed?
You need to do a lot of different, you can't just do one, but you need to do multiple cultures from multiple people. You need to do animals too, and you look at all of it. I agree that no one lab experiment is going to address it for sure.
Right. And I guess, I would think that no one epidemiological model is going to address it either. I mean, he's a proponent of one set of things that he's talked about in these preprints, and there are other epidemiologists with other models that are coming to some of the same kinds of conclusions.
Yeah, I think that's the key thing. If many epidemiologists with many different sort of approaches are coming to the same conclusions, then you can start to say, all right, there's something to these conclusions.
Well, I don't know that the others are dissociating it the way he is, right? So that's maybe what unique to his 2 manuscripts there.
I don't really know that they are, but I don't know that they aren't. It seems to me that that's a general thing that one needs to do. And so it seems logical that others are doing that but perhaps with different models, different dissociation algorithms, if you will.
And they're probably, at the beginning, many of them got something around 2.
Right.
It's not like some got 2 and some got 57.
Yeah, 2 to 3. Yeah.
Right.
And
Right.
so in that sense, you can say, okay, it's 2-ish.
Right.
But I think that the chart that CDC released is not R-naught. It's an RERT, whatever. It's an estimation after, all other environmental, population effects, and so forth. And I don't know what's the basic part of the virus contribution to that, right? And he doesn't know. He thinks there's a percentage. I don't know. He said for Delta, it's 60% more transmissible than the ancestral. So that would be 60% of 2 to 3, I guess, which is not putting it near chickenpox. So I just don't. So I guess I have two issues here. First is how the press relates this. I mean, even Fauci and Walensky, they say, it's as transmissible as chickenpox and more virulent. I just think that's wrong to make those two conclusions. And then the press just repeats it. And we are not sure because these are models, essentially. And I think we need more data. However, Rich Condit would say, but we need to act quickly. Well, what difference does it make? Wear masks and get vaccinated, right? the increase in RTE doesn't matter if you're vaccinated and distancing. So I want for the textbook to know if viruses change in transmission over time, because I haven't seen a good example of that, maybe because we don't have the data. This could be the time that we get those data, but I want to make sure they're right, that viruses may increase transmission over time by X changes in the genome. And I think it's going to take 5 to 10 years to know the final answer for SARS-CoV-2, right?
Sure. I agree with that. But I like his idea that, this is a completely new thing and we're in on it from the ground floor. We have this additional capability of sequencing and getting all this information throughout the whole thing. So yeah, in 5 years, we might really be able to know more about that. It's going to take time.
I think that the press, I think that when we read these things and we think about them, we have to ourselves dissociate. What information does Vincent, the virologist writing a textbook, need? And what information does Vincent, the grocery store patron, need?
Oh, the grocery store needs to get vaccinated and wear a mask.
Right, exactly.
And that's always been the case. So I don't know what difference does it make to say that the virus is more transmissible and virulent, that it evades the vaccines. That's going to make people not get vaccinated, right?
Well, and I would like to think, although I can't go back and I don't have their quotes in my head verbatim, but I would like to think that they said something like, it may be more transmissible, it may be more virulent, and therefore we need to do our standard mitigation of masks and distancing and vaccination.
Yeah, they said it is definitely more transmissible and may be more virulent. That's what she said, yeah. But I don't understand the point of saying either unless you're trying to scare people to get vaccinated. But I don't think that works. Yeah.
Well, it may actually be part of the rationale. I don't think we know exactly what is going to work to get people vaccinated, but I would venture to guess, having heard that, vaccination appointments are on the increase, that it has.
I think what's done that is the uptick in cases, right? In a lot of these southern states, suddenly people are going, oh my gosh, maybe we should be vaccinated. I don't think it's because they've heard that it's more transmissible or perhaps more virulent. I think the cases do it.
But why is there an uptick in cases?
There are 99% in unvaccinated people, right?
But potentially there's more cases because maybe that we have this Delta variant and maybe it is more transmissible.
If everyone were vaccinated, we wouldn't see the cases, though. That's the point. I think in the end, it's just, why don't we get vaccinated? What's the problem?
Because as you heard, it took more than 5 years for everybody to get vaccinated against polio.
Yeah, but those are ancient 1950s. I think we have
the same kinds of things happened in 1918, happened in 1950s, happened in 2020.
Well, I think
you're not going to change human nature, Vincent. I think that,
the problem we have now is that it's become a political issue, and we have one party that wants to make it a election issue, and they're playing with people's health. And so people are not getting vaccinated. They're not wearing masks, which is, other than that, we could do it a lot quicker because we did do 50% of the population in about a year, right? Which is amazing.
Right. But I don't think it's entirely a political issue. I think it's a historical human nature issue that CDC is still trying to figure out. And if thinking that, maybe scaring some people will get some more people vaccinated, maybe that's part of the approach.
I'd be surprised.
The idea of telling people, if you're vaccinated, you don't have to wear a mask. They probably thought, well, that'll cause some people to get vaccinated because they won't have to wear masks anymore. They're trying everything, but they haven't sussed out human nature.
Yeah, and this isn't just a political issue. If you look at the data on the unvaccinated individuals, there are people who are unvaccinated because they can't afford to take the day off work, or they can't afford to potentially have to deal with side effects or childcare. There are other things going on here, and I think that actually understanding them and making sure that we can get people vaccinated and deal with the actual barriers that they're coming across is important.
So you think
that's a whole other story.
Half of the U.S. is in that category because we're at 50% fully vaccinated now. Is it possible?
I don't know what percent is in which category, but I think that if you look at the data, there are people who are who have some reasons for not getting vaccinated that we should figure out how to address. It's not just I hate vaccines.
I think that's part of it, right? We got these 12 people who get paid.
There's some people
say that.
There are some people who think that.
But we have states, as you know, where the governors forbid mask mandates. They actively prevent it. And how can you justify that, right? I don't get it. Anyway, the other issue I have, this is a broader issue, is that whenever there's an outbreak, as soon as they get some genome sequence, they say it's getting more transmissible. Oh, there's just change. Remember Ebola, the West African Ebola outbreak? There was that glycoprotein change, one amino acid, came up early. Everyone was saying more transmissible. And it just bugs me that immediately we jumped to, they, and not me, because I try and be more discriminate. They jump to this conclusion. Why? What's the point? And then the press goes with it because they would love to see an uber killer virus emerge that transmits incredibly. I just don't get it. And that's part of my opposition to this has been from historical epidemics where they end up not being more transmissible. And it's just a mistake, which is easy to make in the heat of an outbreak, I think. And then this idea that subsequent variants are getting more and more transmissible just seems crazy, unless Jeff is right that this virus is really brand new and it's changing, in which case we've missed out on HIV. We missed 60 years of its evolution in people because we only found it in 1980. Too bad we didn't pick it up earlier and were able to sequence it. So anyway, I think it's illuminating. The listeners can decide what they think, but I will await the next 5 to 10 years. It won't go in the textbook until it's proven, in my view. I would like to know what each of those changes mean, what they do in various models, and then we'll see what happens. And then if you think it makes a difference to tell people that I think you need to get vaccinated and wear a face mask and you'll be okay.
I also think that we've all learned that when we look at our textbooks and those tables of the R-naughts of different viruses, we all should take those with a whole lot of salt.
Right. Yeah. As Vincent and I were discussing earlier, it could be that each of those data points for measles and HIV and influenza 1918 and so forth, all of those data points could have come from different people's estimates based on different models, and they might or might not be looking at naive populations. We're comparing apples and oranges.
Well, and they might be taken at different times in different levels of urban setting.
Seasons, all that, right.
I think that this assigning R-naughts to this is really, here's the document. Here it is.
Right.
So it goes from 5 to 8. Mm-hmm. But interestingly, so the original, this is from an original New York Times graph, right? 2/28/2020. And the original has R-naught on it, but the CDC took it off here. Look, they said the average number of people infected by each sick person. So they changed it. Well, this is good. I'm glad it's not R-naught. But it is from 5. So it's not correct to even say it's chickenpox because it might not be. Depends. And I guess these are different circumstances, different scenarios, right?
Different estimates, different ranges of confidence.
So what I want to know
the data
is how much of this is due to spike changes, RNA polymerase changes, nuclear protein, whatever. I want to know exactly that. And then we can talk. I'm just kidding. That's where I'm coming from anyway. Anyway, hope that was useful. Think it was useful? I think so. Mm-hmm. I learned a lot.
I do.
Yeah.
Yeah, the table in the textbooks. You better have a little footnote saying, do you remember Pirates of the Caribbean? So the guy said, it's not really a rule, it's more of a guideline. Yeah.
Yeah.
We should have a little footnote. Oh, boy. Transmission of Delta variant versus ancestral strain. Yeah, it makes you wonder. I want to go back and find the measles data and see how they did that. Do you think?
Well, and if you remember, at least when I talk about it, I say, the R-naught is estimated to be between 12 and 18.
Yeah.
It's not a hard number. It's a range.
Well, I certainly have a better appreciation now. And I will tell students when I show this is an estimate. Yeah. It's a guideline. Yeah. Measles is more transmissible than seasonal influenza. But exactly how much depends on a lot of things.
Right. And that was Jeff's point about that graph, was that the variant seems to be more transmissible. Its block of data is to the right of the ancestral.
Yeah, and my only question is, can you really dissociate it? Because I remember when Alpha arose, they said, it was under lockdown. So obviously the virus was, but so many people emailed me and said, don't believe them. We weren't under lockdown. We were in the pubs every night. So it's like the study, the school study where they said a couple of months ago, Daniel brought this up, what's the effect of having a 3-foot distance from a 6-foot distance? And Daniel said they didn't actually confirm that people were using 3 and 6-foot distances. That was just
right.
Right.
It was what they had on paper, but it wasn't necessarily what they were really doing.
So if you're locking down or you're really locking down, I think that makes a difference. It could make a big difference. So anyway, let's do a couple of emails here.
Before we do that, can we do the PSA about
please.
ASV town halls?
Please do.
So we have 3 of them scheduled from when this episode drops. One on August 12th, Thursday, one on the 18th, which is in Spanish, and one on August 19th. So you go to asv.org/education. This is for everybody. This is for the lay public. Get your questions about COVID-19 vaccines answered to the best of the experts' ability. And if you're out there on the edge of what we know scientifically, we'll say that. We don't know the answers to those things. But still questions to be answered. So sign up.
I want to just point out, I found a nice review article at Nature on a guide to R, the pandemic's misunderstood metric. It's really good. I think it's probably open access. I got it on the train this morning. Mm-hmm. And there's a paragraph which just jives with exactly what Jeff said. RT and R0, R-naught both vary with the social dynamics of a population. Even an easily transmitted virus will have trouble spreading in a region where people rarely meet. In January, COVID-19 R-naught, no, it's not COVID-19, but we'll give you that. In Wuhan was 2 to 3. After lockdown, estimates put the RT there at just over 1. And Kathy, you had a question, has the U.S. ever been to RTE less than 1? And in that article, there's a map which shows in some places it did, right?
They don't show the United States. At least I didn't think they did, but I found another link where you could kind of look at that. But yeah, so it sounds like the effective R or the RT or RE, which now are clearly different by Jeff's definitions, have dipped below 1, but I don't know for how long and over what, when those happened and where they are now.
I think it was also interesting that he said it's really hard to know if it's droplet or aerosol or both. And anyone who said, oh yeah, it's definitely aerosol, oh yeah, it's definitely droplets, are probably wrong, right?
The answer is probably it's all of the above.
Yeah.
It may be. It may be, yeah. All right, let's do some email. Kathy, can you take that first one?
Anne writes, hello, my virologists. I've been unable to find a CDC recommendation about singing again. My course director is suggesting we gather and sing outside. I live in Western Massachusetts, where we have a high rate of vaccinated people. I would really love to get together and sing again, but I would appreciate hearing from you all whether or not we should. In my conversations, I have come to refer to you as my virologists after listening to your podcast for so many months. I'm very grateful to have scientists to listen to. Thank you. So I was deploring this situation earlier in the summer about singing outdoors. Is that okay? Do we need to wear masks outdoors? CDC has been just dreadfully silent about this entirely. I don't have any recommendations. I know that singing with most kinds of masks, or at least the kind that I have, it is hard to take a really good deep breath inside. But I have heard that there are other shapes of masks that are better for singing, that are, I guess, relatively expensive. But if you just use them for singing, that could be a way to go. So depending on how far apart you are outdoors, should be okay. But in Western Massachusetts, it's going to get cold for too very long. So I don't know. CDC has not made any recommendations, and it's disappointing. But would we take the CDC recommendation at face value? It's another issue.
Yes, so I just searched, and a lot of states have guidelines for singing, but they're all several months old, and are, before we got into this recent surge. So.
Right. And with this recent surge, University of Michigan has now said indoors, everybody masked again, regardless of vaccination status.
Yep.
So I think that's going.
Yes, same here.
Did you ever have no masks at Michigan?
Yeah, we did. If everybody is vaccinated, then you don't have to wear masks.
I think we were due to go maskless last week, and now they've changed here also. So, which is fine. I have been always wearing a mask everywhere. In the New York subway and the trains here, you need to wear a mask. So
yeah. But you and I and Brianne are sitting in our offices where we don't have to wear masks, as opposed to my people in the lab are all wearing masks all day long.
Yeah, when I go in the lab, I wear a mask for sure. And
I'm thinking about my plans for teaching classes in masks in the fall.
Yeah, so we're going to teach in person. I guess we're going to wear masks. Yeah. Yep. Brianne, can you take the next one?
Sure. Chris writes, hi, TWiV team. It's a balmy 95 Fahrenheit with a high humidity and a heat index of 101 here in Atlanta, Georgia. As I'm sure you're aware, the CDC has changed its guidance on masking, recommending that even fully vaccinated individuals wear masks indoors in areas of the country where community transmission is substantial or high. This is in response to evidence suggesting that viral loads based on CT are nearly the same between breakthrough infections of vaccinated individuals and the unvaccinated, which in turn would imply that a vaccinated individual is just as capable of spreading the virus as an unvaccinated individual. At least this is my understanding. I don't have any sources to cite, unfortunately, as this information was just presented in an internal webinar. After hearing this, I kept thinking back to what is often discussed on TWiV, where we just don't know how CT correlates to infectivity or viral load. As a humble biochemist focusing on synthetic DNA, I must admit I don't quite understand how this all fits together. Would any of you be able to explain how it is that CT values wouldn't necessarily correlate with the amount of infectious virus in an individual? I think I recall Vincent saying something along the lines of a variant that could throw off more RNA. Would this mean the virus is carrying multiple genome copies, or that it's multiplying faster? In context of CDC's new masking guidance, would it be possible that just because a vaccinated individual has a similarly low CT value as an unvaccinated individual, that the vaccinated individual is not equally able to transmit infectious virus? If that's the case, I suspect the CDC is erring on the side of caution, since, as you all pointed as you all have pointed out, there just haven't been infectious virus studies. Looking forward to your response. Thanks for all you do, keeping us informed and sane amidst the flood of emerging information. Best, from Chris, who is a biochemist. So I was having a similar conversation to what Chris is asking recently. And one of the things that I finally explained to someone was that if there's more infectious virus, that probably means there's more RNA. But if there's more RNA, that doesn't mean that there's more infectious virus. You could imagine that the RNA, which is what we're measuring on CT, is from viruses that are, say, defective, or from debris from dead viruses or things like that. And so I know that dead viruses becomes a whole other kettle of fish, but I'm not going there right now.
Inactivated viruses.
Inactivated viruses. And so with the CT, you're measuring RNA, but you don't know if that RNA is inside of an infectious virus. And so that's part of the problem here. And as we talked about last week, there are also big differences in vaccinated and unvaccinated individuals over time.
Yeah, that's the key. That Singapore study, right?
That's the one.
Right. And way back when, in the beginning, we were talking about RNA levels, and RNA levels would rise, and then they would go down and they would tail off. And there was a long time in that tail off of RNA levels where it didn't seem to correlate with presence of infectious virus. And so that leads to the idea that you could have inactivated viruses, plenty of viral RNA or viral messenger RNA that's not in a capsid where it can safely then be infectious. So.
Yeah. So there always, I think a lot of us have always had this assumption that RNA is so unstable, it's not going to stick around. And so if there's RNA there, it must be inside a capsid. And we're learning a lot more about reasons why maybe that's not so true, and ways that some of this viral RNA may be persisting even in the absence of infectious virus.
Yeah, I think that what you're seeing is the inoculum that went into the nose and maybe reproduced once. There's antibodies there that's neutralizing it. Maybe it's an abortive infection, and then once you get past that first day or so, then it drops very quickly. And so whether that initial inoculum can be transmitted is, I think, highly unlikely. And, the CDC said the other day, I think Rachel said, the vaccines no longer prevent transmission by Delta. And I think that's not supportable by the data that we have. I don't think that's a good thing to say, because it makes people say, well, I'm not getting vaccinated, then what's the point, right? But I don't think we have evidence. And if you really want to say that transmission can happen, you better do some infectivity studies. And I think the Singapore study is really important for that. We talked about that on Friday. And then there is now a study just published yesterday in Eurosurveillance, vaccine effectiveness against SARS-CoV-2 transmission among household contacts. So this, to channel Jeff now, this is a household study, right? He said you have to know the context of your study. I think that I never appreciated. So, a household is different from a supermarket or a stadium or whatever. Anyway, what they find here, this is in the Netherlands, and it is February, May this year, right? But they find, what is the percent vaccine effectiveness that they find against transmission? It's a time when mainly alpha was circulating. So people will say vaccine, I can't find it here. The vaccine effectiveness is they looked at 113,000 adult cases and 253,000 contacts. Wow. All in households. That's a lot of households. And the secondary attack rate, 31% for unvaccinated, 11% if the patient is fully vaccinated. So the vaccine effectiveness against transmission is 71%. And this is for AstraZeneca, Pfizer, Moderna, J&J. They looked at all three. So that's the first study I've seen where they actually looked at transmission by contact tracing, of course, and PCR and so forth. So we need more of that data before we say that the vaccine doesn't prevent transmission by this variant. I don't think that's appropriate. Bothers me a lot. Okay. Andrew writes, thank you so much for the podcast. I'm writing from Atlanta where the heat index is supposed to be 104. Wow, it's the same. Atlanta, it's not the same person, but it's heat index of 101 was the previous emailer, Chris.
It's getting warmer.
Over the last year, the clinical updates have been very helpful for me and my family in dealing with COVID, giving us confidence in the vaccines. The methodical and logical way of presenting research and data, good or bad, is a breath of fresh air from the Facebook doctors and random people with an opinion, and has been a good way to explain COVID and the vaccines to other people. I completed my Pfizer vaccination in March. A week ago, I had a headache for 4 days and some fatigue, figured it was sinus related. Ironically, my 5-year-old daughter was also sick at the time with fever and congestion. I took her for a COVID test, got one myself. She was negative, but I was positive. I've since had 2 negative tests and feel better. The doctor thinks my load was heavy during the time of the headaches. Oh, come on, doctor. And was enough to trigger the first positive PCR test. My question is that now that I've been vaccinated and also had a breakthrough COVID case, do I have even greater super immunity and stronger antibodies? Am I more protected from Delta now than before? Are the antibodies variant specific? Meaning, are they only going to be effective on Delta and less effective on Omega? Thanks again for all your help. Actually, they're good questions. Mm-hmm. So yeah, you got an infection. You got a headache. I know a number of people who are getting headaches. They go for a test and they're positive. So that's one of the phenotypes. It's a clinical sign. It's not a phenotype, right? It's a symptom because you can only feel it yourself. It's a symptom. So I don't know why it's a headache. It's probably cytokines flowing into your brain, right, Brianne?
That's my guess.
Not a low CT doc. All right. So do you have super immunity? Well, you got vaccinated, you got infected, you probably have a really broad response because the studies have shown that if you're infected and then vaccinated once, you get a nice broad response that encompasses all the variants in terms of neutralization anyway. So I don't know if it works in this order, but it might.
But you would expect that, depending on which vaccine you had, well, you said you had Pfizer, so there are 2 doses, and then you've had the virus. So now you've had 3 exposures to this antigen. So you should have boosted those responses and you should have made a pretty high magnitude and also improved quality response. Yeah. Hooray.
They will not be less effective on Delta, and less on Omega. I think you have a broad response.
Right. And when Omega emerges, you will also probably still have very strong T-cell immunity.
That's right.
Which will be effective against Alpha through Omega.
Despite what that cardiologist is saying.
So I always am surprised when I am looking at this because we spend so much time talking about Delta. And I spend some time, I know the Greek alphabet. And so every so often I'll hear about some different one. And I'm like, I feel like we missed some letters. But in fact, we have not missed any letters. It's just that only some of them have risen to the level of being a variant of concern. And others are up to variant of, only variant of interest or something like that. So we are actually up to lambda, at least as far as I've seen. And there is every single, letter from alpha to lambda represented, but only some have actually risen to a level where they are VOC.
Right.
There's a good chart at Our World in Data, which gives you the sequenced variants for the past 2 weeks. The share of analyzed sequences in the last 2 weeks that correspond to each variant. So we have Delta, Alpha, Eta, Iota, Beta, Epsilon, Gamma, Kappa, Other, and Lambda.
Yeah, so I haven't seen a mu yet, but I've seen every letter up to Lambda.
But, in Brazil, Gamma is really 75%. Delta is losing out.
But Brazil is where Gamma
started.
Was first identified. So that makes sense.
But Delta's there, okay? So I would argue if Delta is more transmissible, then why isn't it taking over? Is Gamma more transmissible than Delta? Or is it fitter? I think it's more complicated than the models relate. But, Brian, can you give me the Greek alphabet in order?
I can, but I also will apologize in advance to Theodora.
Well, I could just look it up. I could just look it up if you want.
Alpha, beta, gamma, delta, epsilon, zeta, eta, theta, iota, kappa, lambda, mu, nu, psi, omicron, pi, rho, sigma, tau, upsilon is where I was.
Phi, chi, omega.
Chi, psi, phi, omega.
Oh, very good. Yeah, I was looking at a chart here. I would not have gotten past alpha, beta, gamma, delta. Yep, that's as far as I can go. Nice. So there are 24 letters. But Theodora doesn't listen anymore. It's okay.
Okay. Well, I just felt bad for butchering what I'm sure is the correct pronunciation.
Can we do one more? Is that okay? One more round?
Sure.
Kathy?
Bill writes, thank you for TWiV. I started listening to TWiV for accurate information about the pandemic. I continue to listen to the non-SARS-CoV viruses too. I do not mind when you also talk about other things. When you get into the depths of viruses, a tangent into planes or whales gives my overwhelmed brain a chance to recover a bit. I also do not mind if you get, quote, political. I do object when the politics podcasts or economics or others start talking beyond their understanding about viruses. But I know that I can relax, wait a few days, and get the straight scoop from you. Thanks, Bill.
It's not your brother, is it?
No.
He would have told you, right?
Yes.
I think that's fun. I think most people don't mind the diversions, and we certainly enjoy them. Brianne.
Sue writes, hi, TWiV stars. About 6 weeks ago, I had an RSV vaccine. We seem to have a few trials running here, and I'm one of the guinea pigs for the version of for older people. Like a lot of people, I'd never heard of RSV until hearing about it and the early RSV vaccine attempts on TWiV. When we opened our borders to quarantine free travel from Australia, we imported their RSV, and it's rampant here at the moment. My participation is thanks to TWiV, and thanks to TWiV, I'm not sick right now. Cheers, Sue, and Sue is from New Zealand.
I just looked it up. Volunteer for RSV vaccine studies in New Zealand. Payment, $875 to $1,325. You get paid.
And you don't get RSV. It's amazing.
Wow.
Well, you might if you're in the placebo group.
That's true.
I don't know what kind of vaccine this is. Well,
or if the vaccine doesn't work.
So look it up.
Also true.
For the next one. All right, the last one is from Michael. Dear TWiV-tastics, I was delighted to see Vincent Racaniello on the thumbnail for TWiV 786. I'd worried that I offended Vincent with that graphic of how my Amazon Echo devices parse his name. I'm not. That wouldn't offend me. Other things do, though. Well, not offend, get me grumpy. Anyway, I've got an actual virology question. My son, who was fully vaccinated with the Pfizer vaccine, has gotten sick twice this summer since coming out of his pre-vaccination isolation with some viruses that produce cough, sore throat, and congestion, but by PCR testing are not SARS-CoV-2. When thinking about the coming school year, he wonders whether it would be better to mask at school so as to avoid illness of any kind, or alternatively to stay unmasked so that his immune system will be periodically challenged. He's concerned that isolation over the last year has resulted in an immunoquiescence that leaves him vulnerable to every virus that passes his way. So my questions are, one, is my son justifiably concerned that his immune system will weaken if not challenged by the normal array of pathogens to which the unmasked are exposed in a school setting? And two, even if his concerns are justified, is it best to remain masked when in school so as to help slow the spread of SARS-CoV-2? Thanks for your collective input on this, and thanks for all the great information you've shared with us over the course of the pandemic. I think you should wear a mask, and I think you're getting plenty of immune education even with a mask. What do you think, Brianne?
That is exactly what I think as well. I think that you come into contact with far more microbes in a day than you realize. And so your immune system has plenty of education and plenty to do. And so I would not worry about your immune system being inappropriately educated. And I think that wearing a mask while you're in school so that this pandemic can be stopped is really important.
I like immunoquiescence. That's good.
I think unless you were in a bubble like the bubble boy, I mean, a true, 100% all the time bubble, you're going to have plenty of antigenic stimulation.
Daniel said something good 2 weeks ago. I think he said, people complain that masks are traumatic for kids, but it's more traumatic when a kid is in the hospital. For the kid, that is. Well, probably for their parents too.
I'm pretty sure for their parents too.
Yeah. Yes, it's quite traumatic. So, mask up. I went to 2 different stores this weekend. I wore a mask in both. And one was Home Depot. Masks required, right? Everyone's masked up. It's great. And the other one, they didn't require, there's no sign on the door. Nobody was wearing a mask inside. The other one was a boat store, which I think attracts a different clientele from Home Depot, and they're probably politically different, and they're like, I'm not wearing a mask. So I was the only one in the store. And me and the one checkout guy. And I was waiting for someone to say something. I would have said, hey, back off, man. I'm a virologist. You remember that line from Ghostbusters?
Mm-hmm.
Brienne, did you see Ghostbusters?
I have seen Ghostbusters.
Back
I'm
off,
not
man.
that young.
Back off, man. I'm a scientist. All right. Let's do some picks. Brienne, what do you have for us?
So I have an article that I read recently that talks about giraffes. The article is called Giraffes May Be as Socially Complex as Chimps and Elephants. And it talks about some recent observations about the social lives of giraffes and how they interact. They were originally thought to be completely solitary, aloof, and to not have social groups. And this kind of goes through a lot of research on the fact that no, giraffes actually have a lot of social behaviors and seem to be really social animals. And I thought it was really interesting to kind of read and learn about this, and not only learn about giraffes that are cool, but also how exactly we study this.
And what kind of social behaviors do they have, Brienne?
So they seem to be in groups more often. They actually will sort of stay together among different generations. So it seems like the grandmothers are actually taking care of the baby giraffes in some cases. They will stay by, a body if one dies, they'll stay by it for a long time. And so, I just thought it was really interesting to know how exactly you would study that, to stay why they just decide if a giraffe is social or not.
You got to be sitting somewhere with binoculars and watch them for days and days and days at a time, right?
So they say that the part of the reason they've been able to do this more frequently is because now they have cameras that they can use.
Yeah, that's true.
And to watch them for long periods of time, but also to kind of map the spot patterns so that they can actually figure out which giraffe is which.
I was visiting some university. Where was it? I don't remember where, but I went in the lab and they were studying elephant behavior in Africa, right?
Mm-hmm.
And they had cameras, and the students were sitting there on their computers watching the cameras in Africa, and they're counting how many times this elephant goes here and there, and they don't even have to go there anymore because all the cameras are set up. Isn't that amazing? Field work is revolutionized.
Absolutely.
Giraffes are very cool. I think
they are
awesome.
Very cool. And there's some lovely pictures of them in this article too.
Yes, they're really cool. I remember having, as I've told this story before, we used to have this zoo near us in our little town, and they had a giraffe. And it used to put its neck over the fence, and kids could feed it, and it would just lick out nuts from kids' hands. And I remember our oldest son, he was like 3, and he was giggling that this giraffe was licking his hand with its soft tongue. He's so soft. It's just the coolest thing. Giraffes are cool. Animals are cool.
They are.
I think they're all neat. I come home to deer every night now. My front lawn, they're all there. There's the mom and a couple of fawns, right?
Mm-hmm.
And I get out of my car and I usually go get the mail. And the mom just looks at me and she keeps chewing. So she's looking straight at me and chewing. And then the babies run away. And I'm thinking she's probably teaching them that I'm okay. And eventually they're going to get used to me, right? But the look is really cool. Right. The eyes looking right at you and seeing what you're doing. And if you just walk away, they're okay with that. But the little guys go far away. And then last night, one of the babies went under. It's not so big. I think they're 4 months old now. So they're getting bigger, but they still have spots. And one of them went under the mom to get some milk, and the mom just kept walking and like, nope, time to grow up. No more. Kathy, what do you have for us?
I picked an astronomy picture of the day that's moon of Saturn, Iapetus. And it's a painted moon in 3D. And when you first go to the page, eventually it shows you with a little finger icon that you can move it around. But it's centered on a ridge that they say it's an equatorial ridge that makes it look like a walnut. And so they directed this Cassini spacecraft around it in 2007. And it's pictured now in 3D. And it's just what you can spin it all the way around and see the dark side. But it has these amazing light and dark spots. And the dark spots, they think, are some kind of carbon. Anyway, since we mostly focus on our own moon, I kind of liked this one. And in fact, it has a little bit of a walnut look to it.
Iapetus was the father of Atlas, Prometheus, Epimetheus, and Menoetius. Wow.
Mm-hmm.
Some outstanding kids there.
Yeah.
One year I had a telescope and I looked at Jupiter and Saturn. They came by on different nights and you could see the tiny bright moons because they're really reflecting a lot of light. They're very, very small, but they're so bright that you could see them scattered around each planet. Very cool. The moons were more interesting than the actual planet. Neat. You know, in the early days of the internet, I used to go APOD every day because there wasn't much you could do in the early internet, right?
Yeah. No, I still have it as my browser homepage. So I see it pretty much every day.
Oh, that's a really good idea.
Yeah.
My pick is an NPR article, which I think is important to tell everyone, at least down south. Arkansas governor wants to reverse a law that forbids schools to require masks. So the governor of Arkansas, Asa Hutchinson, he signed a bill months ago banning state and local mask mandates. And now he thinks it's a bad idea. He's changed his mind. In hindsight, I wish it had not become law. And so he's got to try and reverse it now. And hopefully he can. But I think it's a good idea. This guy recognized his mistake and he wants to fix it. Yeah. So Florida and what else? Louisiana, Texas, right?
Yeah.
Please
recognize your folly. Do the right thing.
Yeah, Hutchinson did say that part of the reason he signed it is because he knew his veto was going to be overridden. But, he probably should have made them do that.
Yeah, he should have.
Forced it.
He should have been on record as being against it because that's important. But I'm glad he changed his mind. That's good. Hopefully they can reverse it.
Yeah.
All right, that'll do it for Epitope 792. I'm sorry, it's an old joke by now, but I just love it. It's going to be a while before I stop saying that. You can find the show notes at microbe.tv/TWiV. Send your questions and comments, twiv@microbe.tv. If you like what we do, consider supporting us, microbe.tv/contribute. I went to the incubator this morning before coming up here. I can get out of Penn Station, 2 blocks. I'm there. Oh, it's such a short commute. I love it. And my desk came. I bought a stand-up motorized desk, right, that I'm going to work on. And I bought a ladder because I need to be able to put stuff high up. I bought a little cabinet with drawers on rolling wheels. They were all sitting there. And so I'm going to go back tonight and bring some equipment down from here and slowly move stuff over. But there's a lot of construction going on in the next suite, so I may have to wait before. Because all these suites were empty for a while because of COVID. A lot of companies went out of business, so they're slowly re-renting them. But I'm very excited to go there. Kathy Spindler's at the University of Michigan, Ann Arbor. Thank you, Kathy.
Thanks. This is a lot of fun.
Brianne Barker's at Drew University, BioProf Barker on Twitter. Thanks, Brianne.
Thanks. It was great to be here.
I'm Vincent Racaniello. You can find me at virology.ws. I'd like to thank the American Society for Virology and the American Society for Microbiology for their support of TWiV, and Ronald Jenkins for the music. This episode of TWiV was recorded, edited, and posted by me, Vincent Racaniello. You've been listening to This Week in Virology. Thanks for joining us. We'll be back soon. Another TWiV is viral.